before I start, I would like to thank the organizer for inviting me to attend a symposium. And the other thing is, here I would like to share the experience that we learned from Xylella diseases in Taiwan. So before I start, wait, okay. Before I start, I would like to acknowledge the people who actually did the work. The thing is, Xylella diseases has been in Taiwan for over 20 years. And I didn't join the program until 10 years ago. So the first person who found the disease was Dr. Su Qiu on the Pear's disease of Xylella. And the other thing, the other person who worked on insect vectors is Shi Xianzhong, and both of them have published collaboratively with me on the diseases. And research was funded by the Council of Agriculture and National Science Council in Taiwan. So as other people have mentioned in the morning, that Xylella diseases is pretty common in the Americas. So the thing we know that based on what they have already found, they're being, I don't know how to use it. Okay, yeah, here. Yeah, there are 5 subspecies have been identified. And before I joined the group, we know there are diseases in Taiwan, but on pear and both pear and grape, but we don't know the genetic diversity of the strain before. And as people have mentioned that currently, the Xylella seems to be widespread in the world, including America, Europe, and Asia. And luckily, what we have learned from the story of China and Taiwan, that at least the case being reported in China has not been verified. So the only thing they know is observed in the red grape that shows typical symptom of Pierce disease, but it has not been proved whether the pathogen is really there. However, in Taiwan, we show is contained in Taiwan and the only country in Asia that's been reported with verified cases of Xylella. So how about the diseases in Taiwan? The first one that's been observed is pear leaf scorch disease. In short, is PLS. So it was originally reported in 1993 that Xylella fastidiosa, there was name reported in 1993 that is the causal bacterium of pear leaf scorch disease in Taiwan. And PLS was found in the area where handsome variety is grown. So this is what we know as the tropical or like low altitude pear variety in Taiwan. So pear leaf scorch disease start to show symptoms in early July and stay attached until the leaf fall in the winter. So the dieback of twigs and branches lead to the death of the infected trees within a few years. However, it usually takes more than 5 to 7 years to see a dead tree. So in contrast to the cases that have been found in olive trees or grapes. In low altitude area that's below 800 meters, POS became a limiting factor in pear production in Taiwan. So that's the case we learned. The second disease is Pierce disease that was found in 2002. So in the morning that Mary already mentioned that we report the case in 2012. That's 10 years later after we found this. And there's a reason behind it. And I will tell you about this in a few minutes. It was found to be both table grape and wine grapes are all susceptible. And as a matter of fact, in Taiwan, none of the grape is resistant. So we don't have the resistant cultivar in Taiwan. And scorch symptoms on grapevine leaves were observed at the onset of berry ripening in late May and early June. And affected trees and branches of the grapevine decline and die back in 1 to 5 years post-infection. And I would like to bring your attention to is that in Taiwan, we have 2 crops of grape a year. So there's a summer grape and a winter grape, and both of them are susceptible. So here is the map of central Taiwan where where grapes are mostly produced, including Miaoli, Taichung, and Zhanghua. And as you can see, the redder the color is, the heavier the infection is. So the grape-producing counties and the percentage of PD-positive vineyards are found in all of the in Miaoli and Taichung, but not in Zhanghua. And there is a reason because of the Zhanghua, although it's the major grape production area in Taiwan, but because the high level of underground water, so the plants will be replaced every 10 years. So therefore, at this moment, we still not found any PD-positive strain, a PD-positive vineyard in Zhanghua County. But in Miaoli and Taichung, they are heavily infected. And we have, it takes us 10 years to try to eradicate the disease, and it is not successful. So that's the reason why we finally report the disease after 10 years. So in 2011, we checked 250 hectares of a vineyard, and to find that there are more than 1,200 vines were confirmed to be PD positive, and those are eradicated in the summertime. In the same year, in the winter, we looked another 135 hectares and found 31 vines were infected. So they are also eradicated. However, after 10 years, we still found PD positive vines. So finally, the government say, okay, now you are ready to publish the result. That's the reason why. So after we found there are both PD and PLS, the first question we ask is, since PLS was found in 1993 and PD was not found until 2002, so therefore, is there a possibility that the bacterium was transferred from pear to grape? Because in Taiwan, all of the fields or orchards that grow pear and grapevines are small, and they are closely in close vicinity, so they are close to each other. So the first question we ask is whether they are the same. And after using the ELISA, we found they are actually not the same. They are actually quite different. So based on the results, we have already published this. We found that PD and PRS strain from Taiwan show divergence in service antigens. So the PD strains are more similar to the American Xylella fastidiosa because this ELISA kit was purchased from USA. And also we performed the PCR reaction to show that POS and PD are not similar to each other genetically. So the primers were developed by Dr. Su and found that POS is not similar to the PD strain. So we show that based on the PCR reaction, we predict there are, the POS and PD strains are divergent in their nucleotide sequence, and the PD strain are more similar to the Americans. So phylogenetic analysis also show, using the neighbor joining tree, expressing the relationship of Taiwan Pierce disease strain with the other Xylella fastidiosa strains, using the ITS sequence, and we found that the grape strains are more closely related to whatever has been identified in the States, whereas pear strain is distantly related to the other strain. So this is the third evidence to show that POS strains isolate from the, and the grape strain isolate from the geographic area are not similar to each other. So later, we decide to go on to biochemical characterization, and we performed the cellular fatty acid composition analysis to find that both strains are Taiwan strains, and we found that monounsaturated fatty acids are different. So the Xylella fastidiosa is enriched in the omega-7 fatty acid, whereas Xylella pear strain is enriched in omega-9. So that shows us that the two strains are not similar to each other. So we did a whole genome comparison to show that Xylella pear strain is distantly related to the rest of the Xylella fastidiosa strain. So therefore, we rename this pear strain to a new species. We call this Xylella taiwanensis, well, as you know. Not surprised. So to summarize whatever has been characterized on the bacterial side, we show that the differences in physiological and biochemical properties of PLS and PD strains in Taiwan suggest that the two bacteria have evolved independently. And the result of a genome-wide comparison indicate the PLS strains are divergent from the other Xylella fastidiosa strain. So the PLS strain was reclassified to a new Xylella species, Xylella taiwanensis, in 2016. So after we know that the two bacterium are not the same, we studied in parallel their epidemics and ecology. So this is similar to what other people have reported on the insect vector and also on the alternative host. But considering the specific area of Taiwan, which still remains the only country in Asia that has confirmed Xylella diseases. So we expect that there are not, we will show, or like we know there must be differences in the vector and the alternative hosts. So the candidate insect vectors in Taiwan, there's no the same vectors that have been confirmed to be PD vector in the States. So we start look into two possible candidates. One is the one that the leaf hoppers in Taiwan that can feed on the weeds in the orchard and in the vineyard. So the potential Xylella vectors in Taiwan, including that have been confirmed, including cola, all these are leaf hoppers that have been shown to be PD positive. So we emphasize on the PD because PLS has been a long problem in Taiwan, and PD is a new one. And seems like PD is more emphasized in the past decade. And cola is the one that's commonly found in the weeds and shrubs near a vineyard in Taiwan. And we, Dr. Shi Xianzhong in 2009 report cost postulates to show that cola is the vector. And there's another vector in Taiwan that shows positively correlated with the onset of disease. So we show that in April, the insect is mostly populated in April, and that's where the disease in the vineyard started to show early symptoms. And this study has been done in 2 years, both in 2009 and 2010, we found that the insect population positively correlated with the disease. But this cost postulate has not been performed. The only thing we know is it seems to be explanatory to why the disease can be observed in the early April and late May. So in addition to the insect vectors, we also look into what kind of alternative host can be the reservoir of bacteria. In Taiwan, there's no winter, so basically the plants do not die. However, during the winter time, the plants do, the leaves do falling down. So therefore, there must be some other plants that can serve as the food for the insect vectors. And those being reported, there are at least 94 plant species in the States that have been found to be the reservoir of Xylella. So the food and host plants of candidate leafhoppers and the weeds in the Xylella fastidiosa positive orchard and the vineyards will be the one that we are looking to. So here shows like 5 different host plants of leafhopper in Taiwan, and including beeton, micania, and mulberry. And also there's another one, is Asiatic dayflower. So they are all being confirmed to be the host plants of leafhoppers that's confirmed to be PD positive. And here we can see that those actually, Asiatic dayflower is the one that we have been used to cultivate the insect vectors in the lab. So in the weeds, in the Xylella positive orchards, survey were conducted in 45 PD-infected orchards, and then 238 species. There are more than 3,000 samples of weeds were collected for PD isolation and PCR test. And we found there are at least 5 alternative hosts, including Haemalus and Amampollacensis, Molatus, and this is lollipop climber. And also there's another one, Jerusalem cherry. They are confirmed to be PD positive, and also we've isolated the bacteria from this infected weeds and confirmed by cross postulates by inoculating grapevine, and the grapevine developed typical symptom of PD. So we found those alternative hosts are potential reservoir, and also the bacteria isolated from the host are infective to grapevine. So the PD-positive vineyards are mainly located at the periphery of the grape-producing area or in solitary vineyards. Means that those are readily accessible to the insect vectors from the weeds of neighboring area. So this is where we think this is the hotspot of Xylella PD strains, PD positive vineyards, where we start looking into the other area where the other country or the other foci where there are low incidence of PD. The first one we chosen is Xinse County. And the distribution of PD infected grapevines in keyhole vineyard of Xinse, Taiwan. This is at the periphery of the hotspot. And if you look at this, the color indicates the PD positive vines. And we found there are like Molotos and lollipop climber that located at the periphery of the infected vineyard, where you can see that similar to what Dr. Purcell has mentioned, that it looks like this is where the disease starts to spread out into the vineyard. And all these colored vines were taken off after they were confirmed to be PD positive, but still this vineyard is still not eradicated. I mean, the PD is still there. So the eradication program is not very effective. The second is in the second vineyard was chosen in Nantou County that is also next to the Xinse, that this vineyard is a solitary vineyard. So there's only one vineyard in that location. The rest of the area, including like rice field, tea plantation, and we found that there is one infected host, alternative host is wild grape. It doesn't show any symptoms. The way we found the disease, the PD positive weeds is by like a survey of every single plant in this vicinity. And we found that within the location, you see the color of vines. Those are infected with PD. And this most likely is from the infected alternative host next to this vineyard. So in contrast to the case in Xinse, this Nantou County does not have any infected vines after 2010, so it has been confirmed to be PD-free, and the eradication program is successful. But considering the geographic location of this vineyard, you can imagine why it is successful, because there's nothing else in this location. So in summary of the second part, the leafhoppers are commonly found in vineyards, and pear orchards are candidate vectors of Xylella fastidiosa in Taiwan. And among those, cola has been confirmed to be the vector that transmits PD in the vineyards. And at least 5 alternative hosts has been identified in the field that are in close vicinity to the infected plants. And eliminating the weeds to reduce the population of insect vectors and initial inoculum of the pathogen should be employed as one of the disease management tactics. So with this, I would like to thank you all for your attention, and happy to answer any question if you have any. Yeah, thank you.